Hi, this is Mrs. Horseman, and today we're going to be talking about DNA structure and replication. Before we get started, I just want to make sure that you know what your expectations are for this unit. You need to be able to tell me that the genetic information that is DNA is determined by the order of nucleotides, so the sequence that the DNA is in. You need to be able to tell me why that sequence of DNA is important to the function of the protein. You need to be able to tell me the steps of DNA replication, so how it's unzipped and how it's copied and the whole process. And then you need to be able to describe to me that DNA is replicated during interphase, which is part of the cell cycle. Before we really get into the details of it, I want to give you some historical background on some of the important scientists that helped actually find all of this important information that we know now as DNA and our genes and how that's passed on. The first one is Frederick Griffith, and Frederick Griffith is important because he's the one that discovered something called transformation. And transformation is just the process that part of the nucleotide sequence, so part of the DNA sequence, is copied complementary sequence onto an RNA strand, and that's actually how the protein production is started. The next one was Oswald Avery, and Oswald Avery discovered that DNA actually is a nucleic acid, and that nucleic acid is what stores and transmits the genetic information from one generation to the next. So he's the one that discovered that your genes came from your parents, and those are stored in the DNA that you have in all of your cells. The next one that is listed here is Hershey and Chase. Alfred Hershey and Martha Chase studied something called a bacteriophage, and so I want to show you what that looks like. Those are bacteriophages. So here's a cell, okay, a bacterial cell, and then these are actually viruses. Viruses are not living things. They are just chunks of proteins and nucleic acids and a whole bunch, just basically like robotic machines that are passing on infected genes, okay? So what they determined was that this bacteriophage, which is just a virus that has a loop, you can see that loop right in there, loop of DNA or RNA that's coded with a protein code. And so when they did their studies, what they actually discovered was that the genetic material in the bacteriophage was DNA, not protein. So they helped support Oswald Avery's discoveries. And then there's Rosalind Franklin. Rosalind Franklin is kind of one of the quiet scientists behind the discovery of the shape of DNA. You've probably heard of Watson and Crick before, but Rosalind Franklin was working in a university, and what she did was crystallography x-rays. So they took these crystals and were able to take x-rays, and she took x-rays of these beautiful pieces of DNA. But it didn't look like what the DNA that we look like now. In the meantime, there was two scientists that were working together, Watson and Crick, and they were trying to figure out the shape and structure of DNA as well. So I forget which one, but one of these guys, one of either Watson or Crick, was in Rosalind Franklin's lab, and they saw this picture that looked like basically a gigantic X shape. It had rings around it like this, and then it had sections that kind of went out and in and back out like that. And because of what they had been studying, they actually had been thinking DNA was a single helix. And when they saw that picture, they realized that the DNA was a double helix. So Rosalind Franklin and Watson and Crick all together actually were the ones that determined the structure of DNA, and that it's a double helix. The reason that Watson and Crick get the fame for it is because they actually won the Nobel Peace Prize, because Rosalind Franklin, by the time that prize was awarded, had died, and you can't get the Nobel Peace Prize posthumously, which means after you've died. So the reason, sadly, that she died is because she got cancer, because she worked with x-rays, and at that time they didn't know that x-rays could cause cancer and that you needed to protect yourselves from it. So kind of a sad story. But these scientists worked really hard to determine they knew what DNA was, but they didn't know what it looked like yet. So they're the ones that kind of came up, or they did come up with what the shape of DNA looked like. So what is DNA? We'll talk about DNA and chromosomes and chromatin. They all kind of mean the same thing, okay? Prokaryotes, which remember are bacterial cells, have a single loop of DNA. That means they have one chromosome, and it's all coiled up right here in the middle of the cell, okay? It's a big loop, but it's all coiled up. They don't have very many genes. They don't need them because they're really small organelles, right? Or organisms, sorry. Eukaryotes, on the other hand, have as much as 1,000 times the amount of DNA. Now, some of it determines on how complex of an organism it is, but DNA, no matter what, is going to be packaged as something called chromosomes. And chromosomes are individual strands of DNA, okay? The number of chromosomes is different from each organism. Humans have 46 chromosomes, or 23 pairs of chromosomes. And we'll talk a little bit more about that when we talk about heredity. But what you need to understand is that one chromosome is actually one really long piece of DNA that's coiled up really tightly, okay? You're not actually going to be able to see that DNA unless it's coiled up, all right? So I also want you to remember that DNA in eukaryotic cells is packaged very tightly. Our cells have over 1 meter of DNA in each cell. So a meter is about this big. So in every single one of your cells, you have this much DNA. But it's packaged and coiled up so tightly that it becomes so microscopic and it can fit in the inside of the nucleus of your cell. Crazy. So here is a picture. This is an animal cell. This is an animal cell, and if you see here, this is the nucleolus, but all of this here, it looks like little itty bitty dots and spaghetti noodles kind of all over the place. All of that is the DNA, and when it's unwound like that, when it's all splayed out, it's called chromatin. So if you hear me say chromatin, like what we have highlighted right here, that just means uncoiled DNA. It looks like a big pot of spaghetti inside the nucleus. When it coils up and it forms these X-shaped X-shaped structures, that is called a chromosome, okay? But basically, any of those things are really DNA, and that's what you need to know. All right, so the structure of DNA, I'm sure that you've already seen a structure and you know that it's called a double helix, okay? A helix is just a spiral shape, so a double helix is actually 2 strands wrapped around each other. Those DNA strands are complementary. This means they're not exactly the same, but one strand can be used to make the other strand. They're not identical to each other, but they match each other, okay? So it's like a code, and this one matches this one matches this one, and it goes that way all the way down. If they don't match together, there's a problem, and that's when something like a mutation would happen. So DNA is actually made up of nucleotides, okay? And nucleotides are made up of 3 parts. Here's a phosphate group right there. This is a pentose sugar, a 5-carbon sugar. In DNA, it's called deoxyribose, okay? And that's important. It has to be deoxyribose for it to be DNA. And then here is your nitrogenous base, okay? It's a nitrogen-containing base. And in DNA, there's 4 different kinds. There can either be, it's either adenine, guanine, thymine, and cytosine, okay? So here you can see on this structure, this is a piece of DNA that's been untwisted, so it's laid out like this, and it actually looks like a ladder, okay? So each one of those ladder rungs are 2 nucleotides that are matched together. On the sides here, we have a phosphate group, so that little yellow dot is a phosphate group, and that is bonded really tightly to this sugar molecule right here, so this deoxyribose sugar. So here's one nucleotide. We've got your phosphate group, a deoxyribose sugar, and 1 nucleotide. And its complement, the one that matches it, is going to be on the other side. So here, right here, is the matching or complementary nucleotide. There's a little bit closer up structure. You can kind of see here, we've talked about structure and function and how the shape of something determines what its job is. So if you see here, there will always be a 2-ringed structure that's paired up with a 1-ring structure. And it's actually important that it's like that because that's why it stays uniform and in shape all the way down. If there were 2 2-ring structures, it would kind of bow out like this and then get skinny and then bow out and get skinny. And DNA can't do that. It needs to be pretty uniform. So there is going to be a 2-ring structure paired with a 1-ring structure, okay? We also have, you can see these itty bitty yellow dots, those are hydrogen bonds. So those are what help keep those nucleotides attached to each other, almost like a magnet, okay? Okay, so an important rule, you have to know this person and you have to know what he discovered. Chargaff's rule, you need to learn it and know it. It's also called the base pairing rule. So the base pairing rule says that adenine will always pair with thymine, and those will have 2 hydrogen bonds together, and that cytosine always pairs with guanine, and those will have 3 hydrogen bonds together. Those pairs always will be paired together in DNA. Okay, so you have to know adenine and thymine, cytosine and guanine. And the way I remember it is A and T together actually spell a word, at. C and G are the initials for cytosine and guanine, and they look a lot alike. The C shape and the G shape look alike, okay? So whatever helps you remember, but you have to remember that those go together, and that Chargaff is the one that came up with that, so we call that Chargaff's rule. So what he said is the sequence of bases on one strand determines the sequence of bases on the second strand. Okay, so the way that the nucleotides pair up is based on his rule. So we call that complementary base pairs. And that's just, again, showing you what I just showed you on that last picture, that the adenine is always paired with the thymine and that the cytosine is always paired with guanine.